I'd like to thank organizers for giving me this opportunity to introduce our study of long-read metagenomics using PromethION to uncover oral bacteriophages, which was recently published in Nature Communications. As you know, human microbiomes are of enormous interest, and the two most diverse human microbiomes are intestinal and oral microbiota, which have hundreds of coexisting species. Among them, bacteriophages in the intestinal microbiome have received increasing attention, whereas those present in the oral microbiota have been less studied in only a few metagenomic studies. For example, a study in 2014 reported an alteration of viral composition in subjects with periodontal disease. More recently, I suggested the oral cavity as an attractive system to study coevolution between phages and host oral bacteria. However, these previous studies were based on short read and do have limitations, particularly in assembly contiguity. Specifically, generation of short fragmented assemblies impedes the analysis of genomic context and detection of viral sequences. Recent studies aim to overcome the limitations using nanopore minion long read, one of which validated viral long read metagenomics using mock communities. Another study showed improved assembly contiguity, although error rates are still higher than PacBio. In this study, I for the first time utilized PromethION and HiSeq for studying oral phageome in human saliva and conducted integrated analysis of PromethION and HiSeq data of more than 30 gigabase per sample. After long-read assembly and error correction using the short reads, the number of contexts longer than 1 kb was at least 3,000 across 4 samples, and 50 was on average 250 kb. On the contrary, the hybrid assembly resulted in much smaller N50. Then I conducted classification of the context based on searching ORFs against databases in 2 ways. The first is HMM searching against virome and PFAM databases and prediction of viral context using 6 criteria, which is implemented in VIRSORT. The second is based on velocity searching against NCBI-NR and obtains taxonomy information of each ORF followed by majority vote to assign taxonomy to each context, which is implemented in CAT. This table summarizes the number and percentage of identified phages and prophages stratified by most confident and likely categories. Those not clustered with a viral sequence in the database were considered as novel. Among the most confident phages, novel ones were from 0 to 44%. Among the most confident prophages, novel ones were from 12 to 56%. On the other hand, those clustered with a viral sequence in the database was considered as known, and one example of a high coverage prophage is shown here. This is a good example demonstrating the long-read sequencing improved scaffolding and ability to place a prophage in the proper host genomic context. The length was 78 kb embedded in 674 kb context with on average 440 short read coverage. Taxonomy of this context was predicted to be Streptococcus using the CAT program. This prophage has a genomic region in the middle aligned with 11 kb known viral sequence in the database without the information of predicted host. The 11 kb contains 40 CDS, but they were not enough for the taxonomy prediction. At the end of the high coverage prophage assembled from the long reads, there's a CDS encoding integrase. At the right end of the prophage, there's a CDS encoding enolase, which is a phage receptor. Next, for all known prophages, including this prophage, distribution of differences in length between each of the prophages and corresponding viral sequence in the database is shown here. If x-axis is larger than 0, it means prophages assembled from the long reads are longer than the corresponding viral sequences in the database assembled from short reads. The average was 46 kb. Next, I'd like to explain low human DNA contamination in our protocol. In this study, we conducted DNA extraction suitable for long-read sequencing. In this protocol, the salivary sample is first centrifuged to harvest microbial and human cells and viral particles as pellets, which are then subjected to enzymatic DNA extraction. Sequencing of this extracted bulk DNA resulted in less than 1% human reads. Honestly, this was unexpected. After several months, we reasoned that this occurred because in our protocol, we collected and stored the saliva samples using the OmniGene kit that had the lytic activity for the existing cells, including human cells. Because human cells appear to be more easily lysed than the bacterial cells, the amount of DNA released from the lysed human cells in the supernatant is likely higher than that released from bacteria. Therefore, the pellet contained remaining bacteria and viruses and almost no human cells. In fact, an additional experiment using the RNA narrator stabilization solution with a lytic activity revealed that the proportion of human reads was at least 30%. Next, I focus on top 10% high coverage phages and prophages across the 4 samples with expectedly high error correction rate. A proteomic tree of the high coverage phages and prophages revealed a notable group indicated by the red line. Analysis of the group using a larger proteomic tree, including other reference viral sequences, revealed its location distinctively in the cfOBLD and among Streptococcus phages and prophages. Examination of their genes revealed that 86% of them encoded remote homologs of antimicrobial resistance genes, and 46% of them encoded remote homologs of integrase genes. Next, I'd like to talk about jumbo phages and prophages, which were for the first time discovered in oral microbiota. These figures show the distribution of the size of the phages and prophages. I discovered 5 jumbo phages with genomes larger than 200 kb and 6 jumbo prophages. Predicted taxonomy of each contig is shown here. Except for a plasmid-like element, 9 out of 10 had at least one phage hallmark gene. Overall, there was no overlap in the predicted taxonomies, suggesting they are not confined to specific phylogenetic groups. Regarding the jumbo prophages, they were not previously found among more than 10,000 prophages identified from publicly available genome sequences using DILSOT. Next, I explored characteristics of genes in the oral phages and prophages based on the pangenome analysis. Among more than 100,000 different genes, only 0.3% genes were core and present in all the 4 samples, while 86% were singletons. After functional annotation of the core genes based on HNN, the 2 most dominant annotated categories are phage morphogenesis and host cellular processes, such as transcription regulators. The high fraction of the core genes for host cellular processes after phage morphogenesis supports a recently proposed notion that phages modulate the oral microbiome through multiple mechanisms and represent an additional level of balance required for eubiosis. Finally, I explored the signs of coevolution between phages and host bacteria based on detection of CRISPR spacer sequences in all contexts. This figure shows the number of spacer sequences counted separately in bacterial contexts, phages, and prophages. Of course, the number was largest in bacterial context. When the blood search of these spacers in CRISPR arrays was conducted against all the assembled viral contexts in each sample, on average, 2% of the spacers had homologous sequences or protospacers, which was much smaller than those found in all oral phages in the largest database we use and recently reported in human gut microbiome. It suggests that the oral phages currently present in human saliva could be under selective pressure of escaping CRISPR immunity. I'd like to thank my co-authors and these researchers who made valuable discussions. In particular, Ryan and Nick, who made valuable advice on long-read assembly in the previous ABPHM conference 2 years ago. Thank you very much for your attention.